In this video, we will be discussing the Nirenberg experiments and how they relate to our knowledge of genetics. Our objectives in this video are to discuss who were Nirenberg, Matthaei, and Lieder, what is it that they discovered, the details of their experiment and the procedures that led them to their discovery, and how their conclusions impacted the rest of the scientific community. So to begin, who were Nirenberg, Lieder, and Matthaei? Marshall Warren Nirenberg was an American biochemist and geneticist. J. Heinrich Matthaei is a German biochemist. And Philip Lieder was an American geneticist. They were best known for their contributions to the efforts in solving the genetic code. So what was it that they discovered? They are credited with discovering the first 64 triplet codons in the genetic code. This was achieved by an experiment in which they used nucleic acid homopolymers to decipher the specific amino acids the codons created. Let's begin by discussing the experiment performed by Nirenberg and Matthaei in 1961. They first removed the contents of E. coli cells, this included everything in the cytoplasm, and created an extract with everything needed for the cells but mRNA. In doing this, they were able to completely control the synthesis process of proteins in the cells. After introducing the first synthetic mRNA, U, they discovered that this specific set and order of bases created the amino acid phenylalanine. They continued to repeat this experiment with other mRNA they created, such as AAA or CC. In continuing his research, Nirenberg discovered that not all of the remaining amino acids could be found in this same way, which is where Lieder was added to the team. To find the remaining amino acids, the two used very short artificial RNA sequences, a multiplayer, and a millipore filter. This machine is what's known as a multiplayer multipore millipore filter apparatus. This machine, designed by Lieder, was used by the team to filter out radioactively labeled proteins. Nirenberg discovered that he could suspend proteins in an acidic solution until they precipitated, and then filter out the solution to analyze the amino acids created. The multiplayer allowed them to run 45 samples at once, fast-tracking their discovery of the final triplet sequences. So now that you know how they came to their conclusions, what were the impacts of their study? So these experiments gave us the RNA codon table with all 64 triplet codons. It also created a gateway for other experiments in genetics. These include Khorana discovering how to create synthetic nucleic acids and Hawley discovering the chemical makeup of transfer RNA. These findings also earned Nirenberg the 1968 Nobel Prize in Physiology or Medicine.